I always start with talking about how little fish each person eats every day. So it's only about 60 grams, even though there are about 2.7 kilo of food production per day in the water. So increased economy means an increased meat and fish consumption. And and about 50% of the world grain production goes into feeding domestic animals. Mostly goes to chicken or swine or cattle. And as you heard before many times, we are talking about the blue revolution. I'm not sure if we're there yet or if we'll ever be there, but this is again what we use to get more funds to do our research and to increase the money that we can get out of the politicians. Let's see. Oops. So we have an increase in the farming. So we see that the aquaculture is increasing. The level of the wild fisheries has stagnated for the last 30, 40 years, and aquaculture is increasing. And I will give you a little bit of talk about the world production of the salmon. This is an old slide from 2006, where the total production was about 1.2 million tons. About 600,000 tons in Norway in 2006, and I will show you the numbers from Norway for 2012 as well. So, why Norway? Why salmon? I think that's one question. This is the outline of the country, more or less. It's not completely perfect. I didn't really catch all the borders up there, but we are close to Sweden, Finland, and we have Russian border as well. But most of the border of Norway is, of course, to the North Atlantic. So we have a very long coastline. It's about 25,000 kilometers long. Very few people actually think about that. If we include islands, we have a coastline of 83,000 kilometers. So we have more waters than we have land. It's very temperate. It's quite cool waters. It's a North Atlantic current, so it's mostly ice-free on the coast. And you might not imagine, but the average winter temperature up here is actually close to zero. It's not at the minus. If you go inland, you will have minus 40 or 50. But on the coast, it's still quite warm. And we have very little arable land. So we need to get something more out of it. So if you take the total of the sea and the land of Norway, the productive land in Norway is about 3%. And this is including forest as well. But if you look at productive sea and highly productive sea, we say that this is about 30% of our area. The less productive sea, about 41%. So we have a lot of sea and ocean that we can work with. So it's quite sheltered. We have about 50,000 islands as well, and we have fjords. And we have, most of our aquaculture is, of course, sea-based. So I'm going to take you to one of my favorite places. I live in Oslo, and I was born in Oslo, but my wife was born up here, and we have a farm. So I'm going to take you up there for a little bit of a touristic view. So this is the area. We have a farm out here. This is an island. And close into this, you will see that there are also a fish farm in the area. This is a view from up in the mountain, and you can see there is a salmon farm outside. So of course, one of the things that we have been quite worried about in Norway as well is the environment. How much does aquaculture implement on the environment? One thing is that there is, of course, the feeding regimes. You have the waste for the salmon. How do you actually deal with this? One thing is that a farm can never be in one place for more than a few years. You have to move it. You don't have to move it in this place very far because the current here is quite strong. But you still have to move it every 3 to 5 years, so that the sediments on the sea bottom does not pile up. So just to show you that it is possible to get fish here, this is my wife, and this is John from Scotland, who stayed with us to do some fishing. You can see, even though you have the fish farm here, you can still see the fish outside here, really going up to this on the level of the sea. John is boasting about what he's been catching, of course, and the one he lost, but I caught that one, so he didn't get it. So this is the flatfish halibut. So why again Norway? Why salmon? Of course, we think it tastes good. That's one thing why we want it. And it's quite easy to breed and to culture. And with this, I mean, we know something about the biology, even though we didn't know it when they started some 40 years ago. So how did we do this? Well, one thing is to focus on traditional breeding, which has been very important and still is very important. And the other thing that we did in Norway was to focus on research, and research on a broad scale. It had nothing to do with genomics in the beginning. It had very little to do with genetics in the beginning at all. But it comes. So it started in the autumn of 1971 when people from the Agriculture University collected eggs from about 40 wild salmon strains on rivers in Norway. We do have more than 600 salmon rivers, but they took 40 of the major ones and collected eggs. And they started to construct families from these. It was both full and half-seed families. We tried to keep the environmental variation as low as possible, and difference between families then should be due to inheritance and nothing to do with the environment, of course. And they picked all the starting material from these families created at the time. And the people who did this was a professor of genetics called Harald Skjærvold, and one of his students at the time was Gjedrem, who became the boss of the aquaculture. Harald Skjærvold was the man behind the cattle breeding and the pig breeding in Norway as well. So he made up these institutions. And I think one of the things that we did, which was a good thing for Norway, I'm not sure if you can implement it in Greece or in many other places. It's been done in Sweden and Finland and Denmark as well, was that all the farmers who got the breeding from these breeding stations had to report back everything that happened. Every measurement, every disease, every use of antibiotics had to be reported back. We do it with cattle as well. We do it with pigs. So every animal had a health card. You don't have it on the salmon because it's too many. But for every other animal in Norway, you have a health card, just like the humans. And you have to report back to the breeding station. So it's a collective way for all the farmers, even though they compete, of course, to gain something together that they find is good. And one thing, even though the farmers in the aquaculture as well is competing, they have decided on certain things that they think is on the level above them. For instance, diseases. They're all affected by viral diseases. I'll come back to this. So they decided that, okay, we will do this together. We might not do it when it comes to the breeding for all the various stations, but we can do it at least on diseases and other things like that. So back to the traditional breeding. They started like every other breeding scheme with growth, slaughter weight, age at slaughter, fat and fat distribution with the salmon, and the color uptake. Because in salmon, it should be red. It's actually the most expensive part of the salmon breeding and the salmon farming. But it's no way to get people to buy a white salmon. It tastes exactly the same, but people doesn't want it. They don't think it is a salmon. And the other thing that we did in Norway was to focus on the research. And as I said, it was a very broad approach. One was the physical environment. How can you actually optimize the pens in the sea or the cages on land? How can you feed it? And how do you do the feeding machines? And how is the composition of the feed? This was initially done by the government funds. Now it's companies who do it, so it's much more competition now than it was in the beginning. The management of broodstock was initially also a singular research approach. Diseases and parasites, this is uncommon. And for the last 10 to 15 years, genetics and genomics has also been part of the research initiative done on this. Of course, when it comes to breeding, disease resistance, the growth is no longer important at all in the breeding scheme for most of the breeding firms. But disease resistance is there, and a few other traits as well. So I'm just going to give you what was gained. We're talking about the first 5 generations versus the wild fish. The growth was increased with 113% during the first 5 generations of them. The feed consumption only increased by 40%. FCR went down to 20%. Of course, not all of this was just dependent on the genetics. I know that we say that, but I'm not quite sure if it's true, because at the same time there was a huge impact on the feeding industry and the feed industry. The feed changed a lot during these first generations. But at least it was a success. Of course, at this time, the generation time was 4 years. Now it is less than 2 years. We can push it much lower than that. We can actually make it to the market in less than 1 year. But the cost benefit you have to take into account here, and pushing it a little bit less than 1.5 years makes more diseases, more deformities, and the fish dies in the nets. So about 1.5 to 2 years seems to be an optimal thing there. So just to summarize this again for you before we go into the more genomics, is that this is a very important industry in Norway. It's 1 million tons, or much more than 1 million tons actually this year. And with a value of about 27 kroner per kilo, this is 27 billion Norwegian crowns a year in export revenues, which is about 4 billion euros. We have some unique families and crosses in Norway that we are still keeping. And as I told you, the registrations and the production data is quite unique. We are still able to get them in on a daily schedule. We are routinely performing challenge experiments. We are working very heavily on diseases. I don't know the number of animals used anymore because we're trying to get these down, because the animal rights people are, of course, saying that salmon wasn't actually, or fish in Norway, wasn't said to be an animal until 2004. That was actually when the Animal Welfare Act also took salmon inside it. Before that, salmon or fish was something just swimming in the ocean. You didn't have to treat them well. So now when you slaughter them, you actually now have to sedate them before you slaughter them. And when you're slaughtering 100 to 200 million fish a year, it's a big industry. But we used to use nearly 2 million salmon every year for the challenge experiments. I know the numbers are still around 1 million or so, but I don't have the exact numbers. We do have a large collection of samples from wild and farmed salmon, including pathological specimens, and this dates back nearly 100 years from the wild salmon. So we can go back and we can look into what's happening with the wild fish as well. We have a very large national research environment on aquaculture and marine species, as many of you know. And of course it has, from my point of view, a very interesting biology. It has a duplicated genome. It has a freshwater and a saltwater environment difference. As I'm quite sure many of you know, salmon lives in 2 distinct environments. When it's young and then it's spawning, it happens in the freshwater. And then the older fish go out to the saltwater and migrates to sea to feed. And this is called osmotification. The time it spends in freshwater in the wild is anything between a year and 6 years. In the artificial environment now, we usually take it out of the freshwater around 8 months. It's easy to do it after 4 or 5 months, you can do it, but the death rate of the fish increases in adult life when you push it too much. And earlier it used to be at sea for 3 to up to 8 to 10 years before it went back to spawn in the river. I'm just showing you that this is a very, very big change. We did some experiments nearly 15 years ago now where we looked into specific genes and saw what happened between the pre-smolt, the one in the fresh water, and the one in the salt water. And we found out at that time that maybe something between 25 to 50% of all the genes changes during this time. They either turn on or off, or they change their level. So it's a big, big physiological change in the salmon when it goes from the fresh water to the salt water. So just to say a little bit about what I think the challenges are in the salmon aquaculture for now or for the next time as well. Feed is one of them. The management of the resources is another. SKPs, again, the environmental people are very anxious about the SKPs we have and if they mix with the farmed salmon. So we try to keep the SKPs down as much as possible. We have in the pens in Norway somewhere between 250 and 400 million salmon in the sea at all times. And fortunately, we are down to a few 100,000 SKPs a year, but still a few 100,000 is quite a lot of fish. So this is a big worry. I'll come back to that. And the last one, which is my main issue, would be diseases and parasites. I'm from the vet school, so of course diseases are important in our research. So we'll start with the feed. Oops. There. And of course, one thing is to get the food from the pellets into something looking like this. Of course, a lot of research has been going into the food composition. And as I told you earlier, the feed conversion ratio in salmon is now down to 1.2. But you can also do a lot, of course, on how you distribute the food. This is how it would look like in the old days, where you actually were manually feeding the fish. This is now automatic, so it's all done automatically. And the feeding is optimized so that the fish gets so little feed at the same time that almost no food will go down to the sea bottom. So it's all used before it goes down. This is an old slide, and I had to update with the numbers. So the chicken is much better than it used to be. Now you get 50 kilo of chicken and not 20. You get 85 kilo of salmon. You get about 30 kilos of swine for 100 kilo of feed. And you get, well, the sheep is about the same. Hasn't really changed much. I just made this slide to show you what marine fat has been used for the salmon feeding. If you go back to the 80s, most of the marine fat was used in the hardening and technical industry, mostly for margarine hardening. So this has changed, so we're now using it more and more for animals or for direct for human consumption. So, but what's happening now is that the feeding industry is changing the composition from marine oils to soy. And this is a big challenge. When they started this, even with just incorporating 10 to 15% of soy into the feed, salmon was not happy at all. But during research for the last 10 to 15 years, they're now able to actually make the feed with nearly 50 to 70% of the feed consisting of soy. 50 to 70%. Depending on the feed. But, of course, what does this do to the salmon? Is this salmon still the healthy food as you think it was? Of course it is, and I shouldn't say this, of course, it doesn't have the same level of omega-3 that it used to have. We still say it is enough. It's still a lot. But of course, when you're changing it from marine fat, marine oils, to something from the plant, and salmon cannot make the omega-3 itself, it has to be fed. Of course, the level drops. So it is a level here before you actually say, okay, it's not a fish anymore, it's more like a plant. So I think this is the limit, more or less. But on average, I think the composition for the normal fish will be about 30 to 40% soy in the food. Oops. So I will go and focus on the diseases. So in Norway, we have a lot of viral diseases. There's a list of them here. They are causing or are associated with diseases that are a very great economic burden in the Norwegian salmon aquaculture industry. This is the major threat for the aquaculture. But on the good side, we do have families that are resistant and families that are susceptible to the various viral infections. This is one example. This is the infectious pancreas necrosis virus, where you have some families where, if you look at the challenge test here, 310 families, you have some families where almost all the offspring die, and you have 5 families down here where absolutely no animals in the family die at all. So we do have resistant families, but when it comes into taking this into the breeding, the companies are afraid that if you just focus on these 5 families, you might have a skewed view of what the breeding will do for you. I mean, you might lose some good traits on the side. So they are doing this very slowly. I'll come back to this because this is actually one part where they're using genomics in the breeding. So I will focus on one virus, the infectious salmon anemia virus. And it has losses of more than 100 million euros a year for the industry. It's a double-stranded RNA virus. It's like the human influenza virus. It actually uses the same receptor on salmon to go into the salmon as the influenza virus uses in humans. But of course, there is a huge difference in the immune system in the human and in the salmon. So the virus transmits either with contact with infected fish or through their secretions. Contact with equipment of people who have handled infected fish is also a threat. And the virus can survive in seaweed water for some time. And it's a major risk factor for any uninfected farm if you are close to an infected farm. It also looks like the sea louse can carry them. So it looks like they can passively transform it to the salmon when they attach to the salmon. So what did we do in the beginning when we didn't really know the biology back then? We did divide Norway into regions. This is not the exact region. This is just to show you that we split Norway into several areas. And there is no transfer of fish or material over the regional borders. So we try to contain, if you have an outbreak here, you should actually stop it somewhere here. It shouldn't be going up north or down south. Of course, this is the sea, so you can't really do that, but at least you can try to scale down the effect of the infection. And of course, a pathogen doesn't work alone. It interacts with the environment, it interacts with the host genetic factors. So the whole issue of resistance and susceptibility is where genomics comes in. So we're trying to hunt for important genes. We have, this is another viral disease, which also comes from the infection of the salmon anemia virus. And we I'm wondering what makes the difference between these two fish. Why is one fish resistant and why is one susceptible in the same panel and in the same family? So we try to pinpoint which chromosome it's on, and we might be able to detect that. So we might say, okay, it's here somewhere. But where do we go from there? How do we actually identify the gene when you have the whole area? Because here you will have thousands of genes on it, so you can't really say what is it. And so my main job would be to actually take this area and find out where is the gene and what does the gene do. So how can we use this to get a healthy salmon? If we find this gene, how can we actually make this happy salmon? So how do we identify the genes for the resistance to viral infection? We must have genetic variation. It could be like this, where you have a diseased sibling and a healthy sibling. Of course, the best is to have a large scale of families that show resistance and susceptibility on a gradient scale. And of course, another thing this shows you is that this is a quantitative trait. This is not a single gene. Because if you had a single gene, the families will either be resistant or non-resistant for every individual. So we start out where we need some tags in the genome. We need to find some areas. As Adelina talked about, we're talking about either the sequence. When we started, we didn't have it. So we made all the basic resources: genetic mappings, physical maps, sequence genes, microarrays, libraries of all kinds. And we did this through the Norwegian Salmon Genome Initiative, and the follow-up to that, which was called GENOFISH, or GENOFISK in Norwegian. We were not allowed to put an English name on it for some reason. I don't know why. We have to the research council for that. So we do what Adelina talked about. We generate all the basic knowledge and all the tools that we need. And of course, sorry, there's a picture missing here. It's just because I'm using a Macintosh and this is a PC, so this one always gets away, sorry. We sequence data, we sequence as much as we can for all the functional genes. We don't bother about the complete sequence in the beginning. And of course, we try to use as much data as we can from any other species that we have any information. Then of course the last one is the genome regulation and organization. So when you have this, you can do the QTL studies that Alpino and Kostas talked about. You can find an area where we think there is an interest. But still, this is a wide area. It might be the whole area, or it might be pinpointed down just a few million bases. But still, it's a huge area. But of course, when you have this, you can use this already to implement it in the breeding schemes if you want to. So, of course, since I'm a geneticist, I want to isolate the genes. I want to find out what does this do to the biology. So we're doing functional and comparative studies. We're looking at the microarray, or we're doing other ways of dealing with this. And we're looking at other animals. It might be model animals, it might be humans, if there's a human disease, or mouse or others. So we identify the gene region, and we sequence the whole region to get the whole sequence. And then we can identify genes. But even then, when we have a gene, we have to find out what does this gene do, and is it the real gene, or what is the effect? You might have a single variation in the gene, but the question for you would be, is this still a functional gene product? If it is, it's not your gene. Or is it a non-functional gene product? Then it might be your gene. But you still might not be completely sure about this. So you have to go further. So of course, this is nothing that companies would usually do. This would be for the national science in some way. And this is what we can do for the industry, because we can use basic science resources from the Research Council of the EU to find out the function of specific genes that you are interested in. But of course you need bioinformatics. When I started with genetics back in 1980 and until the mid-90s, you could still do a lot by yourself and by hand. You can't do that anymore, and the more years go, the less you can do. The more you are needing computers and algorithms, and you're depending on this for doing everything. So what we did initially was then to build the resources, and we have used these to look into disease development, heritability of resistance, wild and farmed fish issues, managing the broodstock, gene and genome organization, biomonitoring, and update of the color. So I'm not sure if we should have lunch now. Or?
I think we have to stop,
yeah.
And we start with the Salmon Genome Project after lunch.
Yeah.
If there are questions now for the first part.
Yeah, can I ask you a question? You mentioned that the fats have been changed with soya protein. How you can change the fat levels with soya protein? Can you tell some bits?
It's high. It's
I don't know the exact
40%.
numbers. Okay. Not for this year.
Okay. Do you believe that the increase in fat levels have introduced so many diseases or not in your experience as well?
It doesn't look like it, because most of the diseases came before this change. This change has been done in the feed for the last 5 years, 5 to 10 years. Just because the marine fat is limited, and you can't feed enough marine oil to the salmon for the production you have today. But it is doing something with the fat level in the salmon as well, of course.
I think even in sea bream and sea bass, the feeds nowadays have half of the marine oil protein they used to have. That's consumption.
They follow the same trend with vegetable oils.
We did as an old story, because I think this is the story. I would still do it this way, even though we cannot sequence directly, because I think it's a lot of things to be gained by doing it in a classic genetic way, by building the maps, building the seedling libraries, and etc. So this is a project I was heading for 6 years, because it ended in 2006, we started in 2000. And this was the first initiative by the Norwegian government to put together all our resources in genomics and in bioinformatics at the time, to develop something that could be a national platform for doing genomic research and also building up our bioinformatic resources. Because back in 2000, even though Norway has had a huge software developing industry and huge software faculties at universities to develop things, bioinformatics was very, very small. It was only 2 to 3 people at the time. So the government wanted us to build it up. So this was the first start. Use the salmon to build up the bioinformatics and the genomic tools in Norway. So we did all this, and I will just briefly go through some of the resources before we go into the real issue of this tournament, which is what we're using them for. So when it comes to genetic map, salmon has a few peculiarities. One is that the chromosome number is not constant. If you go into the west coast of Canada, it has 58. If you are on the east coast or west coast of Norway, it's 54 as the 2N number. It's partly duplicated, and it's a large difference in the recombination between the sexes. The male has almost no recombination, the female has a normal recombination rate. So, just to show you that we built the map, there's nothing more to gain from this slide. We used it initially actually for some biology as well. We used it to look into the duplicated region to see how they go through the different linkage groups. The other thing we built up was the back library and the physical map. Again, I will not go into details at all. You could look at the slides afterwards if you are interested. If not, don't bother. We made the library consisted of 330,000 clones. The average gene search size was 190,000 bases. So it's about 19 times coverage of the genome. We made a physical map of this. The fingerprinting was basically done by the Genome Science Center in Canada. We linked it with the genetic map so that we could use this when we actually did the sequencing last year. One thing that was quite important for our research building was sequencing genes. So, sorry, I'll go back here. So this is to sequence cDNAs or part of genes, to get some tags on what kind of genes do we have inside. But when it comes to sequencing, there are some questions. One is to do the sequencing, quite easy. Then you have to verify the results and clean up and remove the bad sequence. I'm sorry this is so, it's not as black as they were when I sent you the slides.
Sorry.
I hope you can see them. You have to find out how many of your sequences are from the same gene. And finally, actually, what kind of genes do we have? These are the 4 questions. So, and the last thing that we did was to find variation in the genes that we sequenced. So, if we start with the sequencing, we sequenced tissues from the young fish initially. We made tissues from both the adult and the young fish, but the Canadians that we worked with made most of the libraries from the adult fish, so we focused on the young fish. So we made them developmental specific and from specific tissues as well. Initially we sequenced 68,000 sequences, and we had nearly 3,000 genes that we were interested in the whole sequence of, that we actually sequenced the whole gene. So the next question is how to clean them. So we developed some bioinformatics tool to process all of the data automatically. And this is a good chance to take out bad poor quality sequences before you actually put them together. So we made a pipeline so that when the sequence came out of our sequencer, the software put them together, took out the good part of the sequence, the one in black in the middle here, got rid of short sequences that were good, and automatically dumped them into GenBank for us. And then they were assembled as well into contigs. Yeah, this is just an example of how it looks when it goes to GenBank. So this is from the eye of the salmon, it has a clone number, it has our SGP number on it. So when you search on this, you will find this in the eye library in GenBank's files. We made various ways to look at this so that we could use it. And the next step was to cluster them, to say how many are from the same gene. So initially we had, well, this is the number that are now as well, nearly 500,000 sequences from Atlantic salmon genes. And we also clustered 300,000 from rainbow trout at the same time. So we put them together and we found that we had a little bit over 100,000 unique genes that we had the whole or partial sequence of. We also looked into the tissue specificity, so we clustered them by tissue or by developmental stage. So the last question is, what kind of genes do we have? So we made, again, an automatic pipeline, and we organized all the genes depending on the molecular function, process in the biology life, or where in the cell are they placed. So if you look at the biology, this is just a test slide. So we had 6 genes that actually goes for behavior. We have in this nearly 400 genes that go for development, and so on. This helps us as a biologist to look into what kind of genes do we have. Or you could look at the molecular function, what has to do with enzyme regulation, for instance, or binding or antioxidant activity, and so on. This is just to show how our website would look like with them, and you can click on all of it, and you can find out which genes are under it. And you can go as deep as you want, and you can check the sequence one by one if you have time to do that. But of course, one of the most important things for me is actually to be able to pinpoint what kind of general terms do I have? What kind of genes are involved in various things? So we did actually put what kind of genes do we have that would be for the immune response or response to stress or response to a virus and so on. Again, this is a test slide that we used initially. Now we have it for all the genes. So we can go in and we can take out all genes that are responses to wounding, for instance, or to virus. And we can work with only those genes. So it helps me as a biologist. I'm not an expert in the computing. I don't want to use a lot of time for computing and finding things out. I just want to pick out, can I get the experts to help me to find which genes do I have that are known to respond to the virus infection? And they can. So it works in a good way. So the last thing was to look for variation in the genes, the SNPs that you know and Kostas talked about, which is a single nucleotide polymorphism. Again, we developed software to do this automatically, but of course, an automatic sequence test will always make mistakes. So if you have time, we go in and manually inspect it as well. You can see here that this is a real change. It goes from an A to a G. So we use these to do some of the science that we wanted to do initially. One of them was the expression analysis. So together with people in Scotland, we made some expression chips. Initially, we made a chip with 17,000 genes, in duplicate, and we made a next generation chip with more than 21,000 genes on it. There was also some Canadian chips available at the time. We used those as well, but I'm not sure if you can see that well, but I always say that ours work much better than the Canadian ones. The European one was much better. I always tease the Canadian with that. But this is not a technology we use anymore. Now Agilent has made an oligoarray that you can actually use for salmon. And we have been going back and helping them to clean out bad sequences and bad data on their chip. So it's now possible to buy it. You don't have to make it yourself. So we have been using this to look into infectious diseases, the infectious salmonella, tuberculosis, and maybe gill disease in Tasmania, and also the IPM. We have also looked into the fatty acid metabolism, how fish take up pollen, and also look into this multiplication, the change from the fresh to the saltwater environment. But we've also been collaborating, especially with some Swedish scientists on using salmon or trout as an environmental monitor because they are in the river. So we have been looking at pollutions in the river, especially on after sewage treatment, because medicines and hormones are not stopped. They go in the river and they affect people living downstream. So we looked for this, the effect of these in the salmon, and we saw quite large effects on it. The Canadians used it to look at the pollution from paper mills, and also used the salmon as a biodetector for that. So the last point I just want to stress is, again, the bioinformatics. And this is what we put up in the beginning. This is 11 years ago, so the world doesn't look like this anymore at all. At that time, I bought 20% of the supercomputer at the University of Oslo, which at that time was a very large one. Now most of us has computers on the desk, which is larger than this. We are now using more small computers in a clustered way. But of course, again, what I spent a lot of time as a biologist and as a project leader was to get the bioinformaticians to do what I want, not what they want to do, but what I want to see. And I want to have things simple. I want to be able to click on things. I want to have things organized so that it's helpful for me as a biologist or for people doing breeding or farming, not because the bioinformaticians think that this is a fun thing to do. And I managed to do that. They were quite good at doing it. So we made several ways of doing searches and queries in the database. And we also in Norway has a setup of 4 supercomputers. This is again an old slide. There's still a network, so whenever we exceed the extent of one supercomputer, there is always another one that automatically will start doing our job. The network is a very high-speed network between all the cities, and it doesn't really matter where we start a job. It will use the available space in any of the 4 supercomputers that we have. Again, this is just to show you some of the things we had there. This is part of the map we had. You could check the details of it. So, what did we use this for? One was to hunt for the important genes, which I will start talking about. The other thing is how we use this for the resource management. It's been used for breeding. And the first thing we did with this was to look for the hunt for important genes. And it tests challenge fish versus controls. We isolated DNA from gills and intestine and did expression studies. Microarray or differential display. And initially, some 10 years ago, we identified nearly 600 genes that had a different expression after infection with virus. So we have also had a fish pathogen working group within the EAGEN Network of Excellence. I headed this group, so we compared our results with the partners in Scotland, in France, Germany, and Denmark as well. So because they did a lot of the same work as we did on different species, some of them, the same virus, and also other viruses on our species. So we tried to collect all the data from all the expression analysis done in all the 5 countries, and looking for genes that we thought was more important than other genes, basic genes for infection and resistance. So we did meta-analysis for all these. And the gene for the salmonella virus has also been fully sequenced, so we can use that as well. So one of the initial things that the Scottish did was to make a QTL analysis, and they found that linkage group 21 had the biggest response to with resistance from this. And this actually coincides, this was IPN, this coincides with some work we did in 1999 on infection salmonella, where we actually find the same QTL region as this. So this seems to be a very important region in the genome for resistant to viruses in general. So we made all these various tests for this, and we ended up having 69 immune-related genes that had to do with this. And we finally, I forgot one slide. And we finally actually found the gene for it. So we have the gene that actually are on linkage group 21. It's mapped, it's confirmed, and the industry is using it now in their breeding schemes. But another important thing is, of course, that the virus itself has also been sequenced and has been used to look for virulence in the virus, because various strains have various virulence, and it's important to find out why and how does this virulence work. So I'll talk a little bit about the research management. The genetic maps and the markers have been used for parentage studies and also to trace populations. We have traced wild and farmed salmon, what happens when the farmed salmon interact with the wild. And it's also being used for tracing food. So I'll just give you a short example of what happens when you're doing parentage testing. We had initially the IPN challenge experiments in Norway, which was quite expensive. And if you remember this slide from earlier, we had all these families with various resistance to the disease. But initially, the fish they had to challenge was very small, 0.1 to 0.2 grams. So there was no way to tag them. So they started out with challenging 30 fish from each family in separate tags. But when you have 400 families, that means 400 tags. And you should maybe do it in double it or 3 times to get the statistics. And then you mean you have 1,200 tanks that you should actually monitor, which is expensive for any industry. So they came to us and said, can you do it with genetics? Can you mix them in the same tank? And can you afterwards use genetics and trace them back to the family and their offspring? So I said, yes, but you have to buy us a sequencer, and I have to employ some technicians. If you can fund that, that's fine. And they said yes. So we developed it, we made the experiment. We had nearly 280 families, 107 males that were mated to 1 to 9 females each. And what we got from them was 6,254 dead offspring. The offspring was collected twice daily in separate tubes and then collected in bags with the date of their death. So we isolated DNA, we took tissue samples from the parents, and we genotyped all the fish with 6 selected markers. So this came to nearly 6,600 fish with the families. So we managed actually to put 87% of the offspring from the parents in our initial run. This is a work we did around 2000, and I had to say to my technicians, who hated this work of course, that they would do something fun after this half a year was done. And as I said, we had a large variation in death rate from 0 to 100% between the families. So this is now routinely used in the breeding scheme of the company. Now we have made other more automatic ways of doing this. But this was the initial experiment we did 12 years ago. So another thing was the effect of the escaped farm salmon on the wild populations. So we did quite a few experiments some years ago. We did 7-year classes of farmed salmon. So we took an average number of fish from 7-year classes so that we had an average genetic background on the farmed salmon. Then we took samples from 4 rivers. This was why we selected these 4 rivers was a double issue. One was that these rivers have been survived or served for nearly 100 years, so that we had samples from nearly 100 years collected. And these rivers were also known to have a lot of farmed fish going up them. But we didn't find any effect at all in the beginning. It doesn't mean there is no effect, but we couldn't find it. There is an effect, of course, of the farmed salmon going up the river. That is that they don't seem to mate with the wild fish. Their instincts isn't as they should be. Very few of them actually go up the river. They just stay around the farm because they're waiting to be fed in normal ways. But those that go up, they do destroy the spawning grounds for the wild salmon. So they don't seem to breed, but they kill off the wild salmon. So the government wanted to have this monitored. So they wanted to look at, one thing was the genetic effects on the wild stocks. They didn't believe our results, so this is work that has been going on for all the years, and we still can't find the effect, but we're still working on it. But they also want to know which farm did the salmon escape from. I think this is basically because the government in some ways think that the farmers, if they have a disease, for instance the salmon anemia, you have to slaughter the fish. And you have to destroy it on land, which is quite expensive for the farmer. So the government in some way thinks that the farmer opens the pen and let the salmon out. I don't really believe that, because the fish that you see from the escapees are not sick. I've never seen, well, I wouldn't say never, but the number of fish that are sick is so small compared to the number of escapees that you can't really think that that would be an issue. So we took one fjord, this is Hardangerfjord on the west coast, and we looked at all the farms, and we also looked at the rivers in this area, this is an area with 50 locations. At that time, this is again back to 2004, 60,000 tons. Now they produce much more there. But periodically they had large amount of escaped salmon. So we tested out initially microsatellites to map them, and it was a great success. We were actually able to say which farm they escaped or the fish that we collected were from. We have developed a more automated set, so that now you can do it with snips and you can automate the whole thing. So as soon as we came out with this, of course, the next time there was an escape from a farm in Norway, we had the ministry on the phone immediately saying that, oh, you have to do this. And we said, well, we don't think we should do it now because we think it's too early to go out and actually do this, even though we think it works. But they say, no, you have to do it. We gave you the funding to do this, now you have to do something for us. So we went up, this was a little bit further north, and we found that the fish was from a specific farm, and they said, no, we haven't had any escapes. Well, we can actually say it's from your pen number 6. So they brought in the manager from last year and said, yes, we did have a few thousand fish escaping. I didn't report it, but I hope that nobody will notice. So it did work. Now it's being routinely used again. So in the breeding, when it comes to the genomics and the breeding, the ISA and the IPN QDLs and the genes that we have found is actually now used in the breeding scheme. I think most of the companies use it. They won't actually give you complete data of what they're doing, but you can go on the website and they will usually announce it as something positive that they will use, because this is a big issue and nobody wants to buy the fish that you could think are susceptible to the viruses. So they're also using genomic selection for other traits. Again, this is used only on the big companies because this is quite expensive. As you noticed with the question you had about this and the farms, I don't think a single farm should do this. It's not. I think it's too expensive at the moment, and it's too risky as well. And as you said, you will have to do it every year. Even though the genotyping goes down and more farms can do this because prices goes down, you still have to do it every year. One thing is, of course, that when you have a specific QTL or a specific gene, you will fix that quite easily, maybe in 1 or 2 generations. Then there will be another QTL that will have to hunt for. So there's no way to stop it. But they are implementing more and more, and they're implementing more and more, especially in the large breeding companies, the genomic selection, where they actually don't look at the phenotype anymore. They're just using SNP genotyping, a whole genome scan, and they're using that for the selection. So it is becoming there. But we have spent 15 to 20 years to get there. So another thing that the genomic results has been used to is to refine and to develop the vaccines. Again, because the virus diseases has been so bad, and we haven't actually been able to develop a vaccine that works efficiently for the viruses. That's why they're now integrating the genomics in the breeding. For the bacterial diseases, the vaccines have been working from day one. But why, for the last 25 years, we have not been able to find any vaccines that works on a good enough scale? I don't know, I can't say. A lot of research, a lot of money has gone into this. Also using genomic data on viruses to predict the virulence. As I said, the various strains have different virulence. So when a farm now has an outbreak of the virus, samples will be collected and the viruses will be genotyped. If it is a low virulence virus, they will usually be allowed to keep the salmon and to sell it to the full market. If it is a highly virulent strain, they will have to slaughter and take them on shore and destroy them. It wouldn't be difficult, I mean, it wouldn't be dangerous to eat it, but we don't want to have them in the sea. So this is used to see see whether you should slaughter or not slaughter the salmon. So I just want to show you a little bit on all the various collaborations we had. The Salmon Genome Project. It started with SalMap, that was the year that I was heading. It's coordinated SalGene. Later on these went into this Norwegian. We had all the Norwegian project that we have collaborated with, Canadians and Scottish. And in a way, all these data has gone into developing the genomic sequence that we now have for the salmon. So the future, in a way, is that we do have a first draft of the salmon genome ready. The length is 2.4 billion bases. It's not a very good sequence, I would say. Number of contigs is nearly 600,000, which means that there are a lot of areas where every sequence we have is quite short. Salmon has a lot of repeats, we also have a lot of duplications that makes it difficult to make the contigs. So the second draft is underway. They say it will be ready in the spring of 2013. So we've used all available sequencing technology, next generation sequencing backends and all these things to make a better sequence. But there's a lot of work that remains regarding the clustering and annotation to find out what genes are, duplications and repeats, as I said. And I have been trying to sell in that we should do more sequencing of genes, and I think that would actually solve these things. But it's been very hard to sell. I've been trying to sell this for nearly 10 years without the Research Council funding me for it. They don't want to listen to that. I'm still doing it with other funding, but I think this will be the way to solve it. So we can use this in a way to speed up our discovery. We can do whole genome scans much more easily when we have the sequence. We can do expression analysis directly on the RNA. You don't have to, when you're using microarrays or other things, you're looking only on the genes you know something about already. With this, we could actually sequence all the transcripts in the genome. We don't have to know about them on beforehand. We are working with microRNAs and with regulation there. We are starting to sniff a little bit on the epigenetics. So I'll just focus a little bit on the microRNA because we constructed quite a few libraries from that. From various tissues, from fertilized eggs to spawning, and from challenged and non-challenged salmon with the RSA virus again. So we identified several hundred microRNAs in salmon, and we have just started to look at the differences between the tissues and the developmental stages. And we're also seeing that microRNA plays a major role in the resistance between the susceptible and the resistant strains of the salmon. So I hope that we can use the genome sequence now actually to look more into the quality of the meat and also into the diseases in the future. So thank you. I'll just show you the acknowledgements at the end here. All the people involved in this. Okay, thank you.
Can I have a question? Can you budget, let's say, with all your contacts and all the present experts, how much it will cost if somebody will go and sequence nodavirus, for example, or try to identify to do an experiment like that? If there are families, we can organize an experiment where we challenge families or groups of sea bass versus the resistance, genetic resistance potential to NODA, because this could be an ideal project for Mediterranean,
yep.
But nobody in the room probably has any idea what will be the process, how to organize it, what will be the resources to commit, and what will be the cost and the time frame for that.
Well, the sequencing will be quite easy. I don't know the size of the NODA virus for you, or
I don't know.
But I mean, still, it will be very, very cheap. I mean, we're talking thousands of euros, and not more than that, usually.
Yes.
So that can be easily done. It can be done in a day or two.
Okay.
Yes.
Amazing.
Depending on how lucky you are. But I mean, that's an easy task. Putting up the challenge experiments, and that takes more time. But, again, it depends what it will cost. As we talked about yesterday, we have this challenge experiment station in Norway, which is partly funded by the EU. And I'm not sure if you still can apply for that, but until at least last year, you could apply as a European country to go there, and the experiments will be paid for.
Okay.
That would be a very cheap way to do it, if it's still possible, I don't know. And this is a containment tree laboratory, so you can do any disease you want, even up to the human diseases.
Okay. Because
but if you're not doing that, if you have to pay, if you go back to my ISA experiments, I mean, we're still not talking large amounts of money. We're not talking millions. I paid somewhere between 10 and 30,000 euros for my challenge experiments. So it depends on the number of families and what you want to do, if you have to pay for it. So I would say, as the worst estimate, let's say a few €100,000 totally.
okay.
I don't know, maybe you have more experience with it.
Because this could be one farm's losses for a year.
Yeah. And as I said initially, I think that even though you are competing, you are farms competing or breeders competing, at least my experience is that when you talk to them, like we did in Norway, I spent some time convincing them, like we did with the sequence as well. It is the farmers who pay for the sequence. Because we said this will benefit all of you. It is not something that you should ask any farm to do or anything. That together, this is an issue for all of you. And if you all spend a little bit on this, it will help all of you in the future.
And you've got a lot of EU projects to support you in the basic science.
Yes, yes.
At the same time.
Yeah, yeah. If we go back, oops, to this one, we had, well, it's actually only two EU. It's the SALMAP and the SALGene for this one. This is the Canadian project. This is the Scottish initiative. And all these others are Norwegian ones, just funded by the Norwegian government.
I have two more questions. One is, do you think that you as an expert could expect some knowledge to be transferred when we sequence, let's say, seabass, if we ever sequence seabass genome? Can we fast track this based on the knowledge of salmon, or can we transfer some kind of, or it's totally different?
Well, the problem with fish is that fish is usually so divergent.
So it's not transferable. Some knowledge is not transferable.
I think it's not possible to be certain.
Yeah.
Not possible to be certain.
They work on it.
Yeah.
Okay.
Because what we discovered, we knew on beforehand that we can't use the fugu, we can't use all the other species. We sequenced cod,
Okay.
not simply, but we can't use it for the salmon because salmon is so different from all the others. Salmon is as far away from fugu or stickleback or the other as it is from humans, so you can just use human sequences as well.
Okay.
We all think that fish, what I usually say to my students is that you always think fish is the same. It looks like the same, but they're so different.
And the other question is what type of how you manage to recognize that this fish comes from that farm and that cage? You had screening of all the farm salmon in all the farms in Norway.
No, well, that was what we told the ministry that we should do before we did this. We said that, okay, we need to know the total genomic distribution of all Norway. But they said, no, we can't wait. So what we did in this case was that we went to every farm and every pen in the area.
So you sampled them.
We sampled. Yes.
Okay.
In one fjord, eh? In one fjord.
Yes, yes.
But this is 60,000 tons of salmon.
Well, this
50,
was,
50 farms, something like that.
yes.
This is a quite an expensive operation to do.
Well, I mean,
It's for-
the sampling is expensive, yes, but the analysis is quite cheap.
The analysis. Okay. Any questions how we can transfer or can complement what we do here with Salmon? Any funny slide that you want to, an explanation? No questions? Okay. Thank you very much.
A lot of questions.
No. Dimitri, ask the question.
You have to ask me now.
Όλοι έχουμε ερωτήματα τώρα, αλλά
If you,
εντάξει, είμαστε λίγο, επειδή είναι πολύ προχωρημένο.
Can I ask
Δηλαδή,
you?
δεν περίμενε κανείς τώρα ότι είχε αναλυθεί το DNA του μισολόγου και μπορεί να...
in English, speak in English.
We have many questions, but right now, this was unexpected for us.
Well,
So we have to think about this, and maybe we'll be more creative if we have a contact.
sure. You will have my email address, so you can contact me.
A question I have is it possible for sea bass and sea bream to find out if two fishes are brothers? Seriously?
Yeah.
Alive fishes or dead fishes?
Yeah.
Alive.
Yeah.
So you completely remove the opportunity to have crossbreeding, yeah?
Inbreeding.
Inbreeding. Inbreeding.
But it was one.
Αντώνη,
Well, it's quite similar to the brown trout and the rainbow trout, but especially the brown trout. Their genomes are quite alike.
So my point now is we have two strains of sea bass, Atlantic strain and the Mediterranean strain. So will their genomics be different, or how far they will vary?
You have to ask the expert on that. I mean, on the average, it wouldn't be.
We learned that the dog has this type of genome, and it could be similar to other animals. From your talk, yeah.
Some specific ones.
I see. Okay.
So there are similarities.
Yeah.
But you can use the similarity
Yeah.
to rocket from.
Okay.
Mapping the strain.
So my
Because
reason was
by
sea
giving
bass
a
is
lot of
one
samples, yeah.
species. Within the species, there may be strains, and how different will be these genomes?
It's
Wide difference.
99.99 similar.
Because for dogs also it's a quite special case, because all the breeds of dogs that you have here, small dogs, big dogs, barking a lot or not, are the result of selection last 5,000 years. It's too recent, all these breeds of dogs. There we have to do with fish that have differentiated salmon with sea bass and sea bream some 40 million years ago. So cats and dogs are closer than sea bass and salmon.
I just wanted to tell you
You
one
see the
thing
difference.
that actually happens. Sometimes the companies get information they knew they wanted or they didn't want. We actually found out when we started this with this one company here, and I won't name them, we found out that nearly 75% of their parentage was wrong initially.
75%?
Yes, was wrong.
Wrong or right?
Wrong. They thought that the offspring was from a certain parents. In 75% of the cases, they were not.
But you used, I think, 6 satellites, yes?
Well, yes,
6 markers.
yes,
And all the finds you were, let's say, potentially find.
yes. Well, for this one, because this was after they changed their routines, because what happened was that, of course, the people doing the breeding knew how important this was. The manager knew, the personally they knew, but the people who actually did the actual work, they took the bags, transferred them to another place. They was listening to the radio. It was cold because they were doing this in the cold room, just concrete buildings, and they didn't really follow up as they should. So for how long this was going on, I don't know, but they were really shocked. And first they blamed us so that we had done something wrong. And I said, we can't do anything wrong because we're doing this anonymously. We used to just say that these are the parents. We have this offspring, which was another experiment. And we said that it all fits. We can't find the parents back.
So you insist on tank instead of parental.
Well, they did, but for this one, this was after they changed the routines. We did it again, so this was a good thing. Now we found out that we can do it, and they do it better than they did.
May I have another question?
Yes.
What do you mean by the term genetic effect? Because you said, that, for example, I think by mentioning about the genetic pollution or contamination, what do you mean by genetic effect? Change in frequency alleles or something else? What do you mean by this term?
Well, if it comes to pollution, we'll find matching controls.
Yeah. But you detect the differences in frequency?
Yes.
Yes?
Yes. And new mutations as well.
I think, Kantam, you had an answer to the question. Antoni?
Well, I have a more general question. You described a very big presentation with many different information there. And then again, I'm thinking of how much weight do you put in the genomics versus the practices that you follow? That from the first part of your presentation, I understand that, and being in Norway, I understand that it has very big. It's very important, you know. Like you mentioned that every 3-4 years you change sites. You know, you move your farms, or you segmented the whole country to different areas, or every time one farmer has some escapees, immediately reports it, or he should report it. So without these practices, how much actual value you believe that you would have from this?
Well, I'm very clear on this. I think that the most important value is on the way we have dealt with it without the genomics up to now. I think for me, when it comes to cost benefit, I can't really give you a basic line on that because I'm a scientist and I do this because I like the science. I'm also working with the industry as well. So if I can help them, I'm very happy to do that. But if they don't see any value in what I do, I will still do it because it's science. And I get the research fund because it's science. It's also because the government thinks that this might help the farmers in the future. And the farmers think that themselves. But now it's basically for the breeders. It's not for the farmers yet. But for the breeders, they've been starting to use the technology in the breeding schemes. And the farmers, of course, will benefit from that if they will have less outbreaks of viral diseases. But if we had been able to make the vaccine 25 years ago, like we did with the bacterial diseases, there would be no point in doing this.
A trust barrier. People believe on methodologies, on farming and good practices, 100%. But and they want to see tangible. They're looking for tangible solutions. And are we in a curve where we are going to see more and more happening now that you finished some basic knowledge about salmon? What is your feeling?
I find it very hard to predict because you don't have the money in an industry like this, like you have for the humans. I mean, a lot of what we do now is based on what's done in humans. And of course, in humans, most governments are interested in investing into the health of the people. So of course, there's lots of money there, but it's a lot of technology we could use that might be helping the industry. But the industry doesn't see it that way yet. I mean, you can personalize your medicine for many animals, but it's too expensive. I mean, you would invest it in humans. People invest it actually in horses. Do you see people investing it in dogs? So it might come down to an industry as well at the end. But when we're talking about vaccination, as I said, we have 200 to 400 million salmons in the sea at all time, and we vaccinate all of them. So it's a massive vaccination that we do. So it's just amazing. I mean, we're doing it in humans, but in humans you are investing in it. You vaccinate people, you have all these nurses and doctors. But industry has to use it. And for the bacterial diseases, they see the use, they invest in it.